Just a little bit of a refresher on some of the terminology associated with protein structure. Remember that amino acids are the individual units or building blocks of a protein, and then the peptide bonds are the bonds that combine those amino acids together. A residue is simply an amino acid that's part of a protein or peptide bonded together. So that's just how you refer to the amino acids when they're part of a protein. All amino acids are going to have the same basic structure. So the backbone of the amino acid is the same. So each amino acid will have this central carbon atom. And then there will be at least one amino group. So over here on the left, you see the amino group, and then on the right is the carboxyl group. So all amino acids have that same basic structure or backbone. Where the amino acids are different and will have their unique characteristics are in the side group, and that's represented by the R here. So the side group is what will have the various characteristics that we'll look at that really determine how that individual amino acid can combine with other amino acids or interact with other molecules. There's different ways that amino acids can be classified. So the first way we'll look at here is the structure of the side chains of the amino acids. And when amino acids are classified in this way, essentially they're looking at what types of molecules are in those side chains. So the side chains are highly variable, from very small to very large and complex. So when we classify by what's in those side chains, it might be looking at, for example, whether there's a hydroxyl group in the side chain or whether there's a certain like cysteine in the side chain or those sorts of things. So basically looking at what that side chain looks like as a molecule. Another way we can classify amino acids is by the net electrical charge. So in this way, we would say overall, does that amino acid have a positive charge? Does it have a negative charge, a neutral charge, or is it a zwitteron, which essentially means it has both a positive and negative charge, but it turns out to have no net charge, but those charges still exist on the ends. Another way to look at or classify amino acids is by their polarity. So in polarity, they would be either polar or nonpolar. And remember that polar molecules are going to be soluble in water, where nonpolar are not. So all of these types of ways of classifying amino acids really is looking at how are they going to interact. I mean, that's really what makes it meaningful to think about the classification of amino acids. Because, for example, if an amino acid has a positive electrical charge, it's going to be attracted to one with another molecule with a negative electrical charge. If a molecule or an amino acid is polar, it would dissolve easily in water. So that would depend, thinking how it could function in the body. That could move around in the blood, where if something's nonpolar, it would need something else with it to dissolve in the blood. So really that's the sort of the core when we think about classifying amino acids, and is thinking about how they will function in the body or how they will interact with other things in the body. Another way to classify amino acids that is particularly relevant in the nutritional world is the essentiality. So by this, we look at whether the amino acid is essential, non-essential, or conditionally essential. So remember, in the nutrition world, when we say essential, it doesn't mean that our body does or doesn't need it. It really is talking about whether we need to get it from outside the body or our body can make it itself. So an essential amino acid is something that we don't have the ability to make. We need to get that through food. A non-essential amino acid would be an amino acid that our body can structure. And a conditionally essential amino acids basically means that as long as we have the building blocks or the precursors to make that amino acid, then we don't need to get it through food. So the chart on this page, or the table on this page, lists some conditionally essential amino acids and their precursor. So essentially what that means, for example, for tyrosine, as long as we have adequate phenylalanine in the body, our body can make the tyrosine. But if we're low on phenylalanine, so we only have enough to do what phenylalanine needs to do just for itself and there's no extra, then we would need to get tyrosine through food. So that's the idea of conditionally essential, and the conditionally essential amino acids are listed on the screen. So the amino acid sequence, basically when we look at the organization of proteins from amino acids, we have primary structure, secondary structure, tertiary, and quaternary structure. So basically it's an increasing complexity of the protein. So when we look at primary structure, that's simply the sequence of the amino acids. So what are the different amino acids lined up in a row? So that's our primary structure. It just tells us what amino acids are in the protein and in what order. As we look at secondary structure, we have some different levels, and I apologize, a few of the symbols didn't come out here in my slide. So this first one is, well, hydrogen bonding is the first, and then alpha helix, and then beta conformational or beta pleated sheet, and I'll put the little beta symbol and alpha helix. I wrote it on top of there, but a little more clear here, the alpha sign. So basically the secondary structure is looking at how the amino acids in the protein start to sort of connect to each other and fold on each other a little bit. So we have just basic hydrogen bonding, this alpha helix structure that I think people are pretty familiar with. You see pictures of that alpha helix, and I'll have a picture on a slide in a minute as well, and then beta pleated sheet. And then there's random coils or turns as well that turn out to be not entirely random, but that's often how they're referred to. So here you have a picture of the primary structure. So the polypeptide chain here at the top, or polypeptide backbone, you'll see that doesn't differ among proteins because remember those, that backbone, so what's shown in red here, so I'll just kind of circle for one amino acid, is going to be the same. So you see one there and one here, and those are the backbones. And then over below those are the side chains or the R groups, what we looked at in a picture before, that's what is going to make each of these different amino acids unique. So it has, there's some different ones listed here. So basically when we look at a protein, the primary structure is just this lining up of the amino acids. And this is represented, again, remember those are peptide bonds that connect those amino acids together into a protein. This slide shows some of the secondary structure. So on the left here, we're looking at the alpha helix. That again, it's a pretty classic structure that you see there, formed by the coiling of the polypeptide chain on itself. So it forms that sort of circular staircase structure. And then the beta pleated sheet, which is on the right, is more of a pleated sheet. So if you think of it as kind of a paper folded on back and forth on itself, would be the beta pleated sheet. And then at the bottom is this random coil that is sort of a little bit more of an unstable structure, but allows more secondary structure in the amino acid. So then after the primary and secondary structure, we get to tertiary and quaternary structure. And essentially this just gives more complex folding or wrapping around, I guess, of amino acids. So when we look at the tertiary structure, we think about how the hydrophobic amino acids are going to kind of move towards the center, attract to themselves, because remember, most often they're going to be moving around in an aqueous solution, a water solution. So those hydrophilic sides need to be out towards the water, so it will move around in that. And again, the electrostatic or ionic attractions, so positive and negative are going to attract to each other. The covalent bonds between cysteine residues will form those disulfide bridges. So all of this in the tertiary structure really has to do with all those characteristics of the side chains, and that will determine how those proteins end up folding on each other in the shape they take. And as they take shape, that's what's going to, again, impact the function. When we think back to lipids, our lipid unit, and lipoproteins, for example, remember lipoprotein, it's a protein that carries the lipid. So how that protein is structured allows it to carry that lipid around and transport it around through the blood. So really, as we get into this more tertiary and quaternary structure, that's going to determine what the protein can do. Again, the quaternary structure at the bottom here just indicates the interactions between two or more polypeptide chains, sort of hooking together or connecting together, interacting in some way. Again, this just shows some graphics of some of the structures, some of the secondary and quaternary structures. So here on the left, particularly the alpha helix, and this 5 to 11 means those are about 5 to 11 amino acids long. This is showing you the end of the last amino acid here, the carbon or carboxy end of the peptide. On the other end of the amino acid, and sorry, I'm kind of writing over myself here, but this other end is going to have that nitrogen or amino end of the last amino acid in that polypeptide chain. So then you'll have different structural elements throughout. So you see some alpha helix, some beta sheets at the top here. So there can be various structures, various secondary, tertiary structure throughout the protein. And then on the right shows some of that as well with some of the other elements, disulfide bonds, hydrogen bonds, electrostatic attraction, various ways that those amino acids can interact to make that protein take its shape. So finally, kind of to wrap up this portion in just a real brief overview of the structure of proteins, this is a sort of cartoon of hemoglobin. Just to give an example, hemoglobin has 4 polypeptide chains that make it up, and the way all of the amino acid characteristics in the structure of hemoglobin and how it folds on itself, and then these polypeptide chains combine to make hemoglobin, so that would be that quaternary structure. That's what allows hemoglobin to actually bond to the oxygen and have a little space for the oxygen so that it can carry the oxygen through the blood. So again, just a real classic example of how structure impacts function. So as you move forward and as you're thinking about, protein structure and all of these the different bonds and the characteristics of the protein, the really key thing is as you move forward and look at individual proteins, realizing that that uniqueness of the amino acids and how those amino acids interact with one another and interact with then other things in the body when we're looking at nutrition really dictates what that protein can do and how it can function.